So let's start seeing some demonstrations of how to do statistical analysis in Python. And let's begin with correlation tests. So I used this code that we illustrated from the previous lesson. If you remember, we took the ViperDB and the ViralZone data sets, and we merged them together into 43 combined genera, and you can see the 5 first. So for each virus, we have its taxonomy, and we have the genome length and inner and outer radius. And because the genome length spans many orders of magnitude, we also take the log of it. And you can see all the results here. And now we may want to ask the question whether we see some correlation between, say, the inner radius to the genome length. So we can plot it, and you can see the result here. And it indeed seems like we get some correlation. I think it's very visible to the eye. But please note that the x-axis is in log scale, okay? So what you see here is quite close. In fact, those 2 points are over an order of magnitude away from this cluster. Okay, but still we see this nice correlation. So to test whether it's significant, we can use a bunch of different tests. So let's begin with Pearson's correlation, which, as you can see, we use here. So you can see that the Pearson correlation between the genome length to the inner radius, which we put here. So here is the genome length, and here the inner radius. We just take the 2 series and put them within this function. We get an R coefficient of 0.35, which is like moderate, and a p-value of 0.02, which is below the standard significance threshold, but it's not like very significant. It could be just, according to this test, it could be just random result. Now, I don't know what you think, but it does seem a little bit strange, because what you see here, it does seem like a very clear correlation. And the question then arises, why Pearson's correlation fails to find it as strong as it should find it? Okay, but then we can note that we have 2 outliers here. And again, it doesn't seem like very strong outliers when we look at log scale. But if we remember that this is a log scale, if we looked at it in the real scale, it would be like really, really far off. It would be like 10 times, 100 times further away than this cluster. So we might think that these 2 outliers mask the results, and we might want to try to filter them away. So you can see here we use standard pandas syntax to take only the points which their log 10 genome length is below 5, namely everything other than these 2 points. And you can see that we filtered out 2 outliers. And now if we worked on the filtered genera, so the remaining 40 records, you can see that now if we use the Pearson's correlation, then we see a much, much stronger correlation. We get a 0.75, which is a very strong correlation, and we get a very significant p-values, like you see here. Okay, but then, of course, the question arises, was it justified to get rid of these 2 outliers in this case? And like, generally, it's a tough question, because there are many considerations for keeping or removing outliers when we deal with real data in real life. Another way to approach this problem is, instead of using, okay, sorry, before that, okay, so a different way to approach that. So we saw that we could calculate the Pearson's correlation between the genome length to the inner radius. But what we see here, again, it's not the genome length, it's the log of the genome length. So you can try, instead of finding the correlation between the raw genome length to the inner radius, to find it between the log of the genome length to the inner radius. So you can see that here we use the log 10 instead of the raw length. And we do it for both the entire dataset and only the filtered genera. And you can see that when we use log 10, things appear much more stable. So you can see that, yeah, so with the raw data, we get, again, a very strong correlation and very significant result. And if we get rid of these two outliers, now that we work in the log 10 space, it doesn't very substantially changes things. So you can see that it's much more stable to work in the log 10 scaling in this case. Yeah.
In this case, you can claim that the relationship between these two traits are something like that.
Mm-hmm. Yeah, so it does suggest that maybe the correlation is not linear, but maybe it is somewhat linear in the log 10 space, which means that you have might have a polynomial relationship in real life, okay?
Mm-hmm.
Okay, but a whole different way to approach it is, again, to use the Spearman correlation instead. And you can see that when we use the Spearman correlation, again, things are much more stable. So we try all four options. So we try with the genome length or with the log of the genome length, and we try with or without filtering out outliers. And you can see again that the Spearman's correlation is much less sensitive to all of these decisions. You get roughly the same result whether you keep the outliers or remove them, and whether you use of course, if you use or not use the log scale, it doesn't matter at all to Spearman's correlation, because Spearman's only cares about ranking. And of course, the internal ranking of the data doesn't change if you use log transformation. So you can see that you get exactly the same result if you use the row length or the log 10 length. You can see it's exactly the same result, which is it's a nice feature of Spearman's correlation, which is not sensitive to the scaling of the data. And it's also far less sensitive to these two outliers. Okay, so we might conclude that in this case, Spearman's correlation might be more appropriate because we don't really have a very good reason to assume linear correlation. And we can conclude with very high confidence that the genome length of viruses indeed seems to be correlated to their inner capsid size. And we established that both using statistics and by just looking at the data. It's very obvious when looking at it that you see some correlation. Okay, so these two components work together to establish our claim that there is a correlation between them. Now, as a matter of syntax, I will say that you have some nice shortcuts within pandas. If you take a data frame and run the core function, which is short for correlation, it will give you automatically an all-by-all correlation matrix. So it will take all the numeric columns. And for each pair of columns, it will give you, by default, the Pearson correlation between them. So it's very nice if you want to make some kind of exploratory analysis, to just look at the data and see which columns are correlated with.
It does not give you the p-value.
No, it doesn't give the p-value, just the coefficients. Maybe it does have an option, I'm not sure. And you can also specify different methods. So by default, it will be the Pearson correlation, but if you give it a Spearman correlation, you can see you get a different, somewhat different values. And for example, unsurprisingly, you can see that the genome length is a perfect Spearman's correlation with the log 10 of genome length, because again, it keeps the ranking. Okay. Now, we may want to also have a concrete model in hand and see the exact trend line that we observe here. So for that, we need to use the stats models package to and to use its OLS model. So that's the common way in which people import the stats models package. Now to do that, what we need to do is first, we need to decide which is the y value. So we take the inner radius to be the y-axis. And we take the log 10 of the genome length to be the x-axis. But notice that we also use the add constant function here, which is very important. Don't forget to use it. If you don't use the add constant, it will force a line that goes through the origin of the axis. So it will not have what is called the intercept. So a linear regression model is typically y equals a times x plus b. And this component, this b, that allows something that doesn't go through your origin will only be given if you add this constant here. So if you don't do that, you will always get a b which is 0, which sometimes it's a reasonable assumption. It's something that you want to enforce, but I think in most cases it's not something you want to assume, and you want to allow it to use any intercept that it is fit. And then to create the regression model, we use sm.ols, and we give the y and the x after adding the constant. We use the fit function to get the regression results, and then on the regression results we can use different functions which allow us to inspect the result of the regression. The simplest we can use is called the summary, which will simply print a summary with all kinds of statistics, which I will not go over here. I will just note that you get a p-value, which is exactly the same p-value that you get when you ran Pearson's correlation under the same conditions. So when we, sorry, it was here. Yeah. So when we ran with the log 10 scaling, the Pearson's correlation, you can see we got the same p-value that you now see here, which is not surprising. I said that Pearson's correlation and linear regression are really tightly connected, and this is really in many ways different ways to look at the same analysis. And importantly, you also get a concrete model in hand, so you can see the actual coefficients. So the constant will be your intercept, your b value, which you can see according to this model is set to minus 540. And the log and the coefficient for the genome length in log 10 scale will be 178. And you can actually see those params by going to the params attribute of this object. And you simply get a series which gives you the coefficients of all the variables which you use in this model. And if you want to plot it, it's very easy. All you have to do is to plot the line, you need to create some x values. So in this case, we want to plot it between 3 to 6.5, which is the range of the data points. So we just create an array with 2 points. Now it's sufficient to have just 2 points because each line is uniquely defined by 2 points. So if you just give the beginning and end, this really uniquely defines the point. And then if you want to get the corresponding y values for this x according to your model, you simply need to use the predict function of the regression results. And here again, you must use the add constant because you want it to match to the kind of x that was used to train the model. So you add a constant to the regression, to this array with 2 values, and then you predict their corresponding y hat value, which will give you the regression y, so this and these values. And then you can just plot it like any other line. So you give the x, you give the y, and you want it to have be a dashed line like here, so you give this style. And then you get this regression line, which on top of that, you can create a scatter plot showing the actual points. So, sorry. OK. So we covered the topic of numeric versus numeric tests. And now let's speak about numeric versus categoric. OK. So as an example for this type of test, let's for example ask a question whether viruses of different taxonomy groups have different genome lengths. And if we want to simplify it even further, the most kind of simple case we can ask is whether what happens if we just have 2 categories. So let's say we just want to compare DNA viruses to RNA viruses and ask whether they have different lengths. OK, so DNA versus RNA is a categorical question. It's a categorical variable. And the genome length is a numeric variable that we want to compare. And then the question simply becomes, one way to formalize it is whether the means of these 2 populations are significantly different. And then the standard test which we can use is called a t-test. So let's see what t-test does. So its input, again, is 2 groups of numbers. And here it doesn't have unlike, so the kind of implementation that you use, if you use the scipy stats t-test int, that's the implementation in Python. Again, you can see that you give it x and y values, which you may confuse with the x and y values that we gave to Pearson's correlation and Spearman's correlation. But here it means very different things. In Spearman and Pearson, it was the two different variables that we used. So we gave it the different axis values for the same data points. Here the x and y are the same variable, just with two different groups. And here you don't have any correspondence between the x values to the y values. These are just two distinct groups, and they don't have to be necessarily of the same size. So you can, it's very legitimate to run a t-test between, say, a group of 100 samples and a group of 1,000,000 samples. And at least in theory, there is no problem with that. You can still compare the means. So the purpose of t-test is to compare the population means of the two groups. And the null hypothesis is that the two populations have the same mean. And so additional assumptions that this test makes is, again, this does assume normal distribution of the means. And on top of that, it also assumes equal variance. Okay, so we assume that we want to compare the means, but we do assume that they have the same variance. I will say that, again, it's fairly robust to both of these assumptions. So unless you have a very strong deviation from normal distribution, and unless you have very, very different variances between the two groups, then it should be fine to use a t-test. But again, if you have strong deviations from these assumptions, this can cause unreliable results. So the limitations of the t-test, again, it only compares the means, and sometimes we can have populations that are different in other ways, and that will not uncover that. And as a result of its assumption, it's considered to be unreliable for small sample sizes. OK, so let's look at the assumptions in a bit more details here. So again, first, like anything else, most of the time, we don't even bother mentioning it. We, of course, assume that all the samples within the two groups are IID, so we have complete independence of the points. We cannot use a t-test if we resample something multiple times. There are versions of t-tests which can, for example, work in the paired case, but that's not the t-test we look at here. A very important assumption is that the two groups are exclusive. It means they shouldn't have any shared samples. So for DNA viruses and RNA viruses, it's fine, because these are mutually exclusive categories. You don't have a virus which is both RNA and DNA. But if you made an amazing discovery and discovered this virus, which has both DNA and RNA genome, and you decided to categorize it to the two groups together, then it would really break your assumption, and you couldn't use a t-test under this kind of modeling. So that's particularly important when you deal with subpopulations. So for example, if we can ask something like, I don't know, are blonde people taller than average? So if you want to ask that, you cannot really take blonde people and then compare them to all people, including the blonde people. You need to somehow make the two groups mutually exclusive. So a reasonable way to do that is just to make a comparison of one versus the rest. So you can compare blonde people to non-blonde people. And then you can run an appropriate t-test, because now you have the two groups that are mutually exclusive. Like we said, we have normal distribution, but not a normal distribution of the samples, which is something people often confuse. Rather, what it assumes is a normal distribution of the means. And that's actually a much nicer assumption, because we have, from probability theory, we have the central limit theorem, which assures us that for large enough groups, no matter what the distribution is, at some point it will become normally distributed. OK, so as a rule of thumb that people very often use, they say that if you have at least 30 samples, under most reasonable distributions, this would be fine. So unless you have a very, very strange distribution, 30 simple samples should be enough. And like we said, the basic version of this test assumes that you have the same variance. Again, this is quite robust. And also a nice feature, which is also supported in Python, you have the Welsh version of this test, which is sometimes called an unequal variance t-test, which spurs this assumption. So if we use this Welsh version, then this will also work for unequal variance, which is nice. OK, like we said, we only compare the means of the two populations, and they can be still different in different ways. And t-tests will not discover that. There are other tests which we can use, for example, the Kolmogorov-Smirnov, which we're going to talk about. But again, t-tests will only work for comparing means. And as implementation goes, again, that's the function we use. What you get as a result is a t-statistic and a p-value. Now, I don't think that the t-statistic is very informative unless you are a statistician. So most of the time, we just ignore the first argument, and we just look at the p-value. And if you want to use the unequal or Welsh version, I encourage you to use the equal var equals false argument, which will use this version and spur this assumption. OK. Another test which is related to the t-test is called the Mann-Whitney U-test. The input of t-test of U-test again is the same input. You get two groups of numbers. But it works somewhat differently than t-test. You can think about it as the non-parametric version of the t-test, because instead of comparing the means, what it does is it compares the ranking between these two populations. So the null hypothesis here is slightly more complex, but it makes intuitive sense. What the null hypothesis states is that it is equally likely that a random sample from one population will be smaller or larger than a random sample from the second population. Okay, so if we take one sample from one population and a second sample from the second population, you will have that the first is bigger than the second, roughly the same amount of times that you have the second bigger than the first. Sorry?
The mean value?
No, the actual value. I mean, now you did it with a concrete sample. So, for example, if you take RNA viruses and DNA viruses, what these assumptions mean that if you take an RNA virus and you take a DNA virus, it is equally likely, unless I give you more information, you cannot know which is bigger. You have 50% chance that the DNA virus will be bigger, and you have 50% chance that the RNA will be bigger.
But the size of the difference doesn't matter.
Yeah, it doesn't matter. It's a non-parametric test that only looks at the ranking of the data. And that's why it's so strong, because again, now it doesn't make any assumption. It only looks at the ranking. So the actual values, they mean nothing. So you can use whatever transformation you like. As long as the ranking of the data is preserved, then U-test couldn't care less. It doesn't look at the actual values, just at the ranking. OK, so it's a very strong test. And I think in many ways it's more appropriate and it's more robust, of course, than t-test. And on an intuitive sense, I think they work pretty much the same. So in most cases, they will give you pretty much the same understanding of the data. So in both cases, if you get a significant result, you can see that in some sense you have one group which is bigger than the other. Just in the case of t-test, it will be bigger in the sense of the mean being bigger. And in the U-test, it will be bigger in a slightly different sense. OK, but on an intuitive level, you will get pretty much the same kind of understanding of. Sorry? It's more related, of course, to the median than to the mean. But again, it's not exactly the median. So yeah, you can think of some strange examples, I think, that you actually, I'm not sure about it. I need to think about it more. But I think it's also not exactly the median. But I don't want to say something I'm not sure about. Yeah? Sorry?
You can use U-test both for mean and for median. Like for mean is to check that they come from the same distribution, and for mean you have to assume that they have a similar distribution in both samples.
Yeah, but again, notice that U-test doesn't care about the mean. OK, so it doesn't care even if you have very extreme outliers, which completely mess up the mean. So you can have one population with a larger mean than the second group because it has a very extreme outlier, but you will still have that the U-test actually goes to the other direction. So you can have, according to U-test, that one group is bigger, and according to t-test, the second group is bigger. So in theory, it is possible to work this way. Of course, in most cases, it shouldn't be the case. Yeah. Yeah, but it has limitations. So the kind of virtue that we spoke about, that it's non-parametric, it can be an obstacle in some cases. So it is, in the end, blind to the absolute values. It cares only about ranking, which in many cases is something that we want. In some cases, it's something we don't want. And we need to make our own judgments of whether it's something we're interested in. And again, like t-test, it doesn't compare all aspects of the distribution. You can have that the U-test will be negative, even though the distributions are different in other ways, maybe. Now the implementation, again, you use the scipy-stats package, and you have the Mann-Whitney U function, which again, you give it the x and y values, like with t-test. But here, for some reason, which I'm not really sure why they made it, in most tests, you have that the default is, of course, the two-tailed p-value, but here, for some reason, they gave the one-tailed as a default. So you need to explicitly state that you want this two-sided p-value, unless, of course, you want one-sided. OK, questions about U-tests, t-tests? OK. And then we have the Kolmogorov-Smirnov tests, which is different than the U and the t-tests. So the Kolmogorov-Smirnov actually does compare all aspects of the distribution. So this test should be used if you are really interested to know whether you have different distributions. Now you have two kinds of Kolmogorov-Smirnov. What you see here is what is called a one-sample Kolmogorov-Smirnov test, which takes as input a group of numbers and a reference distribution. And what it does, it tests whether the group is taken from the reference. So it can be used to check whether, for example, you have data which seems to be normally distributed or uniformly distributed or any other distribution that you think your data should distribute according to. And you can also specify very concrete parameters. So if, for example, you know that, I don't know, the height of people in the general population is given by normal distribution with a specific mean and a specific standard deviation, and you have a different sample of some other population, and you want to see whether those people appear to have the same height distribution as the general population, you can use a Kolmogorov-Smirnov test to compare their heights to the actual values of the general population that you know you expect, and it will tell you whether or not it seems to be the case. Now, it's a very powerful test. Again, it's non-parametric. It doesn't make any assumptions. But the limitation is that you can only reject the origin distribution. You cannot really prove that it's a specific distribution. You can only reject distribution, but not to prove them. OK? And yeah, if you want to use it, just use the scipy-stats KS test. I'm not going to walk you here through the details. If you need to use it, just read the documentation. It's not very complex. But then we have a different kind of Kolmogorov-Smirnov, which I think is even more useful, which is called the two-sample Kolmogorov-Smirnov test, which, again, like the U and T test, it takes two groups of numbers. And what it does, it tests whether these two groups of numbers appear to be coming from the same distribution. So it's a very powerful way to see whether different groups seem to have a different distribution. So the null hypothesis is that both groups are from some population with the same distribution. And again, it's very powerful. It's non-parametric. It doesn't make any assumptions. But its limitation is that it's somewhat hard to interpret the results. So even if you get a positive result, you see that indeed it seems very unlikely that you get two groups coming from the same population. it can be somewhat difficult to interpret what exactly it means, because it doesn't tell you like a t-test or u-test that one is bigger than the other. Rather, you only know that something between these two groups is different, but what exactly is the difference, it's hard to tell. If you want to use it, use the ks2samp function, which again, you give it the values of one group and the values of a different group, and you get a p-value. And yeah, questions? OK, finally, before we go to also demonstrating how to use all of these tests, which deal with a numeric and a categoric variable case, let's also consider the option that what happens if you have more than two categories. So we saw the t-test and u-test and Kolmogorov-Smirnov. All of them work just in the case of two groups, but sometimes we might have categories which, of course, have more than two groups. We can have, for example, days of a week where we have 7 categories. And also in the example we gave, we may want not only to compare DNA and RNA viruses, we want to compare all the different groups. So we want to compare a single-stranded DNA to double-stranded RNA, and all the different options. And then we cannot really use all of these options that we mentioned. And so we need to take a different strategy. So one option is just to run, say, multiple t-tests, for example, in a one versus all fashion. So we can each time take a group and isolate it, and then consider how it compares to all the different groups that we work with. That's a valid strategy to use, but it does have very strong, it does have very strong limitations, because we then, for example, need to use multiple hypothesis testing, which would really reduce statistical power. So instead of gaining power from the fact that we have many categories, we only lose from it if we go through this strategy. Yeah, question? Okay. So another approach is to use what is called ANOVA, which is analysis of variance. So the kind of ANOVA that we are going to learn is called the one-way ANOVA, where what you get as an input is, again, now you get not just two groups, but k groups of numbers. You can get as many groups as you want. And the purpose of this test is to compare whether the means of all the k populations appear to be the same, or whether you see some difference. So the null hypothesis would be that the means of all k populations are the same. And then if we reject the null hypothesis, we can conclude that at least one of the groups seems to have a different mean, that we do indeed see some correlation, association between this category to the variable that we're looking at. And additional assumption, again, it's a parametric test, so we do assume a normal distribution of means, like in a t-test, and we do assume equal variance, like in t-test. But again, like with t-test, it's fairly robust to these assumptions. So unless you have a very small data set or very, very extreme distribution, it should be fine in most real-life cases. But limitation, again, like we saw with Kolmogorov-Smirnov, that we had some difference, but we couldn't really attribute it to something specific. Here we have a similar kind of limitation, that even if we get a statistical result, then we won't know which is the group that has a different mean than the others. And to do that, we need to use what is called post hoc tests, which are more complex, and we won't cover in this lesson. But it will only tell you whether there is some effect. It won't tell you exactly which effect. So that's the downside of these kinds of tests, which are sometimes called omnibus tests, which can only tell whether there is an effect, but they won't tell you exactly which effect. The implementation in Python that we're going to see is the f1-way function, which works in the same way like t-tests. You just give, instead of two groups, you give as many as you want.